Well, thanks a lot to the organizers for allowing me to come. I really enjoyed this morning. I attended my first Biochar conference at Iowa State University in July, and I sure learned a lot there. I should have learned a lot about how I should change my slides with regard to char and biochar, but that'll come lately. But I'd really like to start by saying that now it's time for something really different. And the older members of the audience may recognize that quote from Rocky the Flying Squirrel. Not that old, I guess I did. Anyway, this is really different. I'm going to talk about a wet processing method. It's called hydrothermal carbonization. And in the thermal processing of biomass, again, I attended my first conference in July at Iowa State to learn about this. And what I did learn is that basically in torrefaction processes, it's a relatively long period of time, at least hours. And you're increasing the energy content by at least 30%, but that's about it. And you're starting primarily with wood or something that's relatively dry, because as a comment was made earlier, to remove water evaporatively is an expensive process in terms of energy input. And that's how hydrothermal processes differ considerably, because we take great pains to keep the water present in our system. The reactions are conducted in water. They actually require water, and I'll show you a slide that actually gets at that point a little later. Many of the reactions are actually exothermic, and it's possible in a continuous reactor potentially to maintain reaction temperatures due to the exothermicity of the reaction. And the energy contents are generally elevated more than 50%. The products are collected by non-evaporative methods in terms of removing water. Gases, we simply, with heat recycle, cool the reaction system and collect them by distillation. Liquefaction produces oily products that can phase separate. And hydrothermal carbonization, that I'll talk about today, gives a filtered product, a filterable product. I call it a char. I probably should have called it a solid, residual solid or something like that. But it's a biochar, as we term it. And we save energy by not removing water by evaporation. And in terms of hydrothermal processing, there are at least 3 different kinds of methods that have been examined in the literature, and some of these are quite old. Carbonization dates back to about 1913. But gasification is done in very extreme conditions, under very extreme conditions, up to 700 degrees centigrade in the absence of catalyst. With catalyst, ruthenium is a common one. The temperatures can go down, but you get mixtures of gaseous products and a fair amount of carbon dioxide. When you're making methane and carbon dioxide, methane has a high heat of combustion, about 24,000 BTUs per pound, but it's diluted by an inert material that doesn't oxidize. That's carbon dioxide. So you have to take it times the mass percent of the heat of combustion of this renewable natural gas, as it's called. And it isn't always that great, but it has its application with certain substrates. I don't think microalgae are among them. Liquefaction is kind of in the middle. You get a phenolic, oily, rich material that can phase separate and be easily isolated. A lot of work has been done in Europe. There's a gentleman by the name of Marcus Antionetti, and he's at the Max Planck Institute in Potsdam. And he's really been the father of this whole modern era of development in hydrothermal carbonization with lignocellulosics. But with these materials, you're dealing primarily with the chemistry of lignin, hemicellulose, and cellulose. We were the first ones, we believe, to work with a relatively low cellulosic biomass source, that being microalgae. And we published our work in the June issue of Biomass and Bioenergy. So it's relatively new work. The conditions are the mildest. We generally work at around 200 degrees centigrade, mild pressures, and the char is an easily filterable material. We had a really good group of scientists and engineers that have worked on this program at the University of Minnesota, and I want to acknowledge my colleagues. And for me, this is kind of like a dream project. I spent 34 years as a chemist in a central or corporate research laboratory of a company doing technology-driven research primarily. And there you come up with a chemistry that can be relatively complex, and then you spend most of your time going out to the business units trying to force fit it into certain applications. It rarely worked. And I was always envious of process engineers that would have a process that people would actually line up at their door and come and see them rather than us hitting the pavement with our pinstripe suit on trying to sell something. But the process is about as simple as you can possibly imagine, and therefore more commercialable. At least it has more commercial potential. You simply take a biomass substrate. We're working with low cellulosic material. A biomass substrate in water and heat it up in a confined system. You get two products initially. You get a char, which we isolate by filtration, and you get a filtrate that has value. And I'll tell you about a third product today, which are the oils or the lipid-derived products, the fatty acids that are present in many of these biomass substrates. The approach that we desired to utilize was to increase the carbon to oxygen ratio, which is a process that is referred to as carbonization generally, by dehydration, by losing water, rather than by working at higher temperatures and splitting off carbon dioxide. We don't need any more processes that generate carbon dioxide. When you lose carbon dioxide, you're losing carbon as well as oxygen. We thought that was deleterious. And with more gaseous products, you're creating more cost and complexity in your reaction equipment because of the higher pressures. We selected microalgae as our first biomass substrate because if you're interested in CO2 capture, and we certainly are these days, per unit of mass, pound per pound, microalgae are unparalleled in their ability to capture CO2 photosynthetically. And they grow at just phenomenal rates, 3 to 4 hours during growth phases. And there is kind of a budding algal oil industry out there. The real promise of microalgae, according to many people, is their fatty acid content. I read one report where and you don't have to use arable land with microalgae, but if you do, there was a microalga that had 70% lipid content, and to accomplish 50% of the transportation needs of the United States, you need 1% of the cropping area. The next closest terrestrial plant is oil palm, which would use 24% of the landmass of the cropping area. So, and the aviation industry is really interested in the lipid makeup because it makes a high energy fuel suitable for jets and aviation fuel. So there are a lot of pluses going with it, but there are a lot of negatives too. Economies of scale is just going to kill them for a long time, and there are some other drawbacks that I'll mention as we're going. But from a practical standpoint, we'd like to eventually process these things continuously, not just in a batch reaction mode. Their small size, just being a few microns in dimension, is conducive to this type of processing operation. You can get reaction temperatures up very quickly with high surface area, small-sized objects. And the chemistry is entirely different. With algae not having a lot of lignin, hemicellulose, and cellulose, you're dealing primarily with the chemistry of proteins, lipids, and carbohydrates. So that was exciting to me as a chemist. And you look at what's reported, and I'm not going to get into this, but if you look at these reactions, there are dehydration reactions that are occurring, which we want to have happen in our system. Where's that? Is this the laser pointer? The red thing? Oh, there it is. This doesn't work very well on my white back slide. But in the upper left, there are dehydration. But if you get a sugar, there are reducing sugars, those that have aldehyde groups present. It turns out, regardless of which sugar you start with, they will transform and eventually all generate these kinds of reactions. In the lower left quadrant, you can get all sorts of fragmentations, rearrangements, eliminations, all kinds of things going on under these reaction conditions that I'll talk about. And proteins and amino acids can even be involved with reducing sugars in this really relatively familiar reaction. We all are familiar with this because this is what happens in the caramelization process in cooking. These are called Maillard reactions or Browning reactions. And they give rise to a whole host of heterocyclic compounds that give rise to the flavor and the aromas that we're familiar with in cooking meat, for example. But when we get these filtrates out of this, because of these Maillard compounds, these generally small heterocyclic molecules, small molecules, there are hundreds of compounds present in this filtrate system. The reactions I'll talk about were all done in a batch mode. This is a stainless steel reactor, 450 milliliters in size, not large. We'd love to scale this up at some point. and get into a continuous mode. But all the reactions I'll talk about have been done in this reaction, which is a reaction system, which is purchased from Power Reaction Systems in Moline, Illinois. And we get great data out of this. And with a microalga that we worked with a lot, this is the Nalliella salina. We buy this from a Chinese source, Alibaba Inc. And it's an interesting microalga in that it doesn't have a cell wall. It has a cell membrane, and it's grown in salt water. So you're not worrying about land usage in this instance. And it also can be nutriated such that it can make up to about 50% oil. So it's really a decent candidate for the algal oil industry. It was grown in mass in Australia in open seawater ponds for a long time, and it's raised now as a source of beta carotene, which is a yellow pigment nutraceutical kind of a material. And what was nice about it also, aside from the other things I mentioned, is that we could buy 5 kilos of it. And I'm a physical scientist, and when you introduce some sort of biological component, variability in your data just goes crazy. We didn't experience that so much as long as we were dealing with one lot of material. So we got really good data out of this. Our standard deviations in the percent carbons and mass yield categories were less than 1%. So what these data show in this reaction space, and we're looking at a reaction space in terms of temperature variation, time variation, and percent solids of the microalga in water, just no catalyst, just straight water. The darker regions refer to higher levels of percent carbon recovered in the algal char. And when you plot these three variables in twos, in the upper right is temperature versus time. These are contour plots. These are really relief kinds of maps of the reaction space. And when you have a lot of lines, a lot of different color variations going on, there's a lot happening with those two variables. Not so much with temperature and time. But in the bottom two plots, when you get percent solids, which is the y-axis in both of those, things are happening very quickly. As you go up a percent solids, the percent carbon recovered in the char is going up significantly. In the, this might show this time. In this, over here, you see we tend to lose it. Likewise, when the time is too long, we tend to overcook the system a bit. And that suggested to us that we could run this reaction at the lowest reaction temperature and reduce the time. The shortest time that we worked with in this study was 30 minutes. We went down to 15 minutes. We felt that was about the lowest we could go in a batch process. And we made more or less the same thing. So we're pretty convinced that we can make this continuously, but that would be a very, very important point to actually demonstrate in terms of its commercial ability. Okay, what about the char products? Now, the comment was made about Hugh McLaughlin earlier. And Hugh, I met Hugh at Iowa State, and he heard a talk that I gave there, and he said he would look at our chars. And I was glad that he did. I would have changed my slides, but I didn't get the data back until recently. But he sent me back the data, and he said, well, it's more of a char, and it's not a very good one. So this is not great in terms of carbon sequestration and things that you're more familiar with, but it's a different kind of a char that's obtained, and we think it has some applications maybe that might also apply to the materials you're making. So it is a char, but it hasn't had the time to form the aromatic structures that give rise to a lot of the properties that you're seeing in the kinds of biochars and chars that you're more familiar with. But here visually we can see, these are at the same magnifications. With natural coal, in kind of a macroscopic view on the left and a microscopic one, you can see it's basically a collection of rocks. And at best you can say microscopically it sort of has a flaky appearance. At the bottom, we looked at a monocultured prairie grass that we had in our possession. We milled it and sieved it and got it as fine as we could to be closer to the algae in terms of actual size. And we still had to treat this for 190 degrees centigrade for 17 hours in order to get any effective level of carbonization. And the image on the left is not changed at all, really, visually from the starting material. And that's because cellulose is present and remains in this material. It's like rebar. and it holds the thing together. And microscopically, again, the surface it's carbonized somewhat. It's more flaky, whereas the algal char has a much higher surface area, and it has a more coral-like structure. I have images of other materials that are actually better than this alga. In terms of properties, with the levels of carbon that are developed under these conditions, what's amazing is we're making what Mother Nature required about 1 million years to make in terms of the natural process for making coal. We're doing it in 15 minutes. And to me, that sounds like it ought to have some value at some point. But the carbon levels that are achieved, typically with algae, we're starting at about 45% carbon. And in 15 to 30 minutes, we can go up to 65 or approaching 70%. There are kind of 3 or 4 positives with these data, 1 negative. The ash content is low. Note the algal char has an ash content, the one from Denali Salina at 0.3%. Very high heats of combustion, higher than the natural coal that we compared it with, which is a Powder River Basin coal that our XL Energy in Minnesota utilizes. The sulfur content is low, but we do have some nitrogen content. The protein content of algae is high, and it drags. I'll tell you a little bit more about this process in a minute or two as well, but it drags the protein and the nitrogen in the char as well. Now, when we talk to coal burning companies, they aren't very concerned about this. They say they could replace a portion of their natural coal. They could supplement their natural coal with this char and could handle that with their scrubbers easily, but it's still something that we need to be aware of. Early on, somebody asked me, well, why don't you just burn the algae? And I didn't have an answer, and I do now. If you go through the comparisons of a scenario where you obtain a wet slurry of the algae, and here we're dealing with Chlamydomonas reinhardti, which we make in our own laboratories, and it can be isolated in 10% solids by centrifugation. So we're starting with both scenarios at 10% solids. And you go through the process, the problem with getting 1 pound of dry alga, of dry microalgae, is you have to take 10 pounds of slurry and get rid of 9 pounds of water. That's 1,100 BTUs per pound of water removed. And note that the heat of combustion of the dry clammy was 7758. The heat of combustion in the char, on the other hand, is almost twice that. It's 13,577, even though we get it in 40% yield and are comparing it 1 pound with 0.4 pounds, go through the old energy calculations, we end up with a positive 2,000 BTU improvement in terms of the energy output relative to input, an overall 4,000 BTU difference in this case. I mentioned that we're trying not to generate CO2 in this process, but we're trying to carbonize things dehydratively, or by dehydration, I should say. And if you go through the carbon accounting, we start with about 8 grams of carbon in the microalgae, and then we end up with about 55% of carbon in the char, about 45% in the solutes, not carbonate. There was no carbonate detected in this filtrate, but they're present in heterocyclic compounds that are formed during this process. In this headspace where the gas would be and whatever would be dissolved in the water, and if we assumed all the pressure, all the gas that was in the headspace was carbon dioxide, at most it could be was 7%. So we are not carbonizing by loss of CO2 in this process at 200 degrees centigrade. You really got to get up above about 240 or so before you start to see really elevated levels of carbon dioxide being obtained. And significantly and importantly, I think, 80% of the nitrogen that was residently present in the microalgae is present in the filtrate, primarily as ammonium ion, and 100% of the phosphorus. And we think that's useful to recycle back into algal growth. And the algae are expensive to grow because they require so much nitrogen. They require about 3 times the nitrogen as does a typical terrestrial plant. So in terms of the first product we can get out of this, and I've put this kind of in a reverse order of where things are, you get, when you talk about energy products right away, you always get pigeonholed right into combustion as being the thing you got to do, and you got to compare it with. And that you'd like to get there, but sometimes higher value added applications along the way can help build the thing and create markets and create a little revenue early. That's really what we're striving to do. So especially because of its low ash content, we think it has promise as a substrate for synthesis gas to convert into industrial chemicals or gasoline. This would be carbon sequestration light. Somebody mentioned you could throw it down a mine shaft. Yeah, that's what we could do. Kurt has looked at this a little bit in terms of soil amendment, and it is microbially active. And if there's any interest in the data, Kurt hopefully could respond. But these materials are interesting in the sense that they're basically hydrophobic cores with hydrophilic groups around them that make them water wettable. And the group present on the surface are carboxylic acid, they're hydroxyl, they're aldehyde, a lot of interactive groups. And these things can interact with themselves, or they can interact with concrete possibly to reinforce it, with polymers. With polylactic acid, we've studied that a little bit with a professor at the University of Minnesota. It does show some reinforcing properties. Possibly as a carbon or a coal coke replacement in steel manufacture might be considered. These last two we do know are not just speculation. They're great adsorbents, and I'll show you some information along that application line, possibly for water and air purification, or as a carbon neutral supplement to natural coal for electrical power generation. The other product we obtain at this point is the filtrate. And as I mentioned, this is a very complex solution. We gave up early on trying to isolate usable, valuable compounds out of that. Not that they aren't there, they probably are, but it's just such a complex mixture that we decided to abandon that route. But if you look at it, the solution itself is nearly neutral. pH is about 6.13 with chlamydomonas. There's a lot of potassium, nitrogen, and phosphorus present in this system. So one thing we were interested in, and we've just done a few experiments, these are not, they're more supportive of the concept than they are in any conclusive manner. But we fed it back into chlamydomonas. And we found that addition of about a 20-fold dilution was equivalent to about half of the optimum nutrient for chlamydomonas. And it satisfied the requirement that it could be utilized as an algal growth medium. We're not sure if we could replace the other half or replace just half the TAP medium or one of its components. We're going to be studying that in the near future, but at least we illustrated the possibility of its being utilized in that concept. But microalgae are hard to get a hold of in large quantities. The whole industry is geared toward making nutraceuticals. Right now, it's in a transition period, I think, where more and more companies are going to try to make high lipid-containing microalgae, but that's something that yet has to happen. And yet there is a material out there that is a very large industrial commodity, and these are distillers' grains, and associated with the ethanol manufacture from corn in the United States. And 23 million metric tons were made in 2008, certainly a large commodity. And one problem with that is that what you get from the centrifugation process, which is you remove the alcohol by distillation from the beer well, and you're left with a mixture of a solid and a liquid. The liquid's called thin stillage. The solid is called wet distiller's grains. Virtually every one of these ethanol plants in operation have centrifuges. And they can centrifuge this material and get a solid that has about 35% solids, 65% moisture. That's called wet distiller's grain. And that's great. You can feed that, and you can truck it to local animal feeding locations within about a 60-mile radius. But beyond that, you got to dry it, or the microbes will get after it, and it'll rot on you. So that it costs a lot of money for that industry to dry that product. And we thought, what about using that 35% solids material and variations on that? What does hydrothermal carbonization have to do with distillers' grain? We did exactly the same experiment that we did with the Denali Salina, and you can see that the process itself is overall more forgiving. It's a wider reaction space that's tolerable in this process with this biomass substrate. It was more tolerant of high temperature and longer reaction times. And the chars are almost identical. I can kind of send these two chars around if I can pass these around. The one with the label on it is from Denali Salina, and this is from distiller's grain. You just pass those around, and they're virtually identical. The only thing different about the distiller's grain char is it has about half the nitrogen. That's because we're starting with less nitrogen. It's not as much a proteinaceous biomass material. Visually, these are all at the same magnification, these SEMs. And you can see that, especially in this, what happens apparently during carbonization under these hydrothermal conditions is components are liquefied, they're solubilized, they carbonize in solution, and they come back and reform in whatever solid in the system to form this kind of a coral-like structure that has a relatively high surface area. I think that image shows it. I've got one more image I'll show you later on that even better depicts this. But to tell you what we found out about distiller's grain is that if you're going to be worried about the energy input in terms of input-output in terms of a combustion situation, 35% solids actually favors burning the distiller's grain. Distiller's grain has a heat of combustion that's pretty respectable, about 9,000 BTUs per pound. We can get it up nearly 13, but at 40% yield, when you start out with something that has a 35% solids composition, actually removing that water is not that big a problem. So but we learned something from this. We learned that actually the sweet spot for us to work with microalgae or with whatever biomass we have is at about 5 to 15% solids. If you're below 5% solids, you're heating too much moisture or too much water up in the system as well, unproductively. But if we're between 5 and 15%, that's where we want to be. So we're continuing to do our investigations with distiller's grain by trying to improve the yield. If we can get the yield up above 40%, that would help things. But we're also looking at the thin stillage, which is at about 7% solids. And that I'm not going to talk about that today, but that holds some promise, I think. And, of course, we'd like to look at this char as well for higher value kinds of applications, as I mentioned earlier. But working with distiller's grain allowed us to kind of relax a little bit and look at some of the fundamental tenets of hydrothermal carbonization. And the reason for that is microalgae, in terms of composition, their protein, lipid, and carbohydrate makeup are all over the place. Not so with distiller's grain. It's a relatively consistent material. We had a lot of it. So one of the first things we examined is this concept from the literature, at least, that water is required for hydrothermal carbonization. So we replaced water with isooctane, which boils conveniently to 100 degrees centigrade, which we wouldn't have to do that, but that worked fine. Subjected it to our kind of our optimum reaction conditions at 200 degrees for 2 hours, and we get a reddish brown char out of this, at 82% yield when corrected for moisture of the starting distiller's grain. The char analysis showed that hardly any carbonization had occurred. We went from 48% carbon in the starting distiller's grain to 50, and I think that was due to residual moisture present in the system. And in terms of mass balance, when we filtered off the isooctane supernatant, we got 13% of lipids that are present in distiller's grain out. So we have a nice mass balance of 95%. So indeed, and remember that what we accomplished under equivalent conditions with water was a conversion from 48% carbon up to about 65. So yes, water is required. The other thing I wanted to examine was whether or not cellulose is totally unaffected at 200 degrees centigrade. Because they do, with lignocellulosics, they work over long periods of time, 16, 17 hours, to get effective carbonization. So what I did was I subjected microcrystalline cellulose, which is it's a colloidal material, and we recovered that in 71% yield after the 200 degrees 2-hour reaction time. There was no carbonization that occurred at all. So this indicated to us that some of the cellulose was solubilized slowly over that 2-hour period at 200 degrees, but it didn't either carbonize and it didn't reform on a cellulosic scaffold. And that may be important for a mechanism and char formation as we move forward. We were also interested in examining the components of distiller's grain singly and in combination, and empirically looking at how char is made. You know, what reactants give rise to char. And if you look at distiller's grain, it has the coefficients of variations for these various components are really pretty tight. It's basically 30% protein, 10-11% fat, 4% residual starch, and 42% of neutral detergent fiber, which is the lignocellulosic content of the corn plant. There's about 19% cellulose present in the distiller's dry grain. So the first thing we did was to apply a literature procedure for obtaining the neutral detergent fiber. It's an extraction procedure where you boil it with a variety of surfactants and solvents and obtain the neutral detergent fiber. But when we tried to rehydrate this, we wanted to go to 42% and really mimic that of distiller's grain. We couldn't really go higher than about 7.5% and get something that was homogeneous and a nice slurry that you could transfer and work with. So we modified our conditions, and while we're at it, we thought whenever we're going to look at a combination of a starch and protein, we'd use cheese whey. We'd done a lot of work with cheese whey. We were told that Wisconsin is having a heck of a time with all the cheese whey. And they make 20 billion pounds of cheese whey a year, and they don't know what to do with it. Well, that isn't quite true. We get a great char out of it, but when we checked with cheese manufacturers in Wisconsin, that was a story that has changed over the years. That used to be a problem in Wisconsin. Their DNR is very strict, more strict at least in Minnesota, in terms of its disposal, what they do with it. But now, because of the lactose content of cheese whey, the pharmaceutical industry uses that as an adjuvant in capsule medications. So they take all the cheese whey and process it that they have. But anyway, if you work with cheese whey, the lactose and protein combination, which is constant for our study, you get a 30+% yield of a beautiful char at 65% carbon. So we used the cheese whey whenever that combination was applied. Lactose was our starch, and you can buy whey proteins at a health food store that are lactose free, which we did. And then we used for corn oil, which is a polyunsaturated fat, and canola oil, which is a monounsaturated fat. And what we did, again, was to examine each of these singly and in all the combinations. There were about 15, 16 different experiments to look at all of them. And what we found is that the triacylglycerides, the fats, do not contribute to char yields. And in fact, were actually located on the char. This was pretty significant. And we knew that because neither the corn or canola oil, when microemulsified at 40% solid, subjected to 200 degrees C, 2 hours, no char. Protein, with and without oil, got the same yields as long as we extracted the char that was obtained. Similarly with the lactose, the starch, and with the neutral detergent fiber. The oil did not contribute to char yield. What it did do, and this was surprising to us, that this suggests that there may be a catalytic component to these triacylglycerides, either as solvents or the fatty acids that are created hydrolytically under these conditions are surface active agents that could form micelles and emulsions that could facilitate carbonization. But with the lactose by itself, under these reaction conditions, we only got about 54% carbon in the char. When the corn oil was present, it went up to 69.5. Similarly, even the protein was carbonized under this instance, from 38.2 to 54.3 with the canola oil. Again, no change in the yield, but the level of carbonization was increased. We also learned that proteins gave char. They contribute to char mass, but by a different mechanism. Typically, they do not carbonize. They just simply denature. They fall out of solution and are insolubilized in that process and are contained in the char. And this was evidenced by the protein lactose yield being greater than the combination of either the protein and the protein and the lactose char, which was 22%, so 30. And also, more significantly, the protein lactose char had a lot of nitrogen in it, about 3.9%, with no lactose nitrogen at all. Similarly, the same thing was observed with the neutral detergent fiber. This is visually indicated, I think, in this image. The one on the left is the lactose. Again, you can see the kind of grape-like cluster, coral structure of spheroids that are coming together to form this filterable char product. Whereas on the right, the protein, it just fell out of solution. Not much was going on with the protein by itself. So what this suggested was a process whereby we could get the fatty acids out. The fatty acids are the algal oil that industry is seeking. And right now they're using a variety of methods that some of them dry, where they dry the microalgae to obtain the lipid products, which just kills them in terms of an energy input standpoint. So we'd conduct our hydrothermal carbonization reaction at 5 to 15% solids, so that if you wanted to combust the char, it would compare favorably with in terms of an energy output situation. We would filter the mixture to obtain the filtrate and the char products, which contain the majority of fatty acids adsorbed on that char. The pH was about 6.13, I mentioned in a previous slide. The pKa of a fatty acid is between 4 and 5, so we'd like to be below 4 to make sure everything's in the carboxylic acid form and not carboxylates, which are not extractable. So we acidified it and then extract the fatty acids from the filtrate. We obtain one product, which is a recycled, which is used to be recycled into algal growth. We extract the char with an organic solvent from the filter or filtrate and obtain the most of the fatty acid products from the char product. I'll show you some data in just a minute. Then you get the extracted char product, which retains a healthy heat of combustion. We lose some of it because the lipids, the fatty acids, are gone, but we still retain about 90% of its heat of combustion. And the fatty acids are obtained, which can get into the petrochemical feed. In terms of the requirements for microalgae, again, we needed a large quantity. We purchased 5 kilos of denali L-acetylene. That was great because we got excellent data and we could do repetitive experiments to change reaction conditions using the same starting material. That's a problem in this. It has been a problem for us. We'd like to examine microalgae that are representative of the industry in terms of their lipid content. And right now, most microalgae growers are just are selecting strains of algae that grow rapidly, that can imbibe CO2 quickly and convert it into biomass. They're supplementing ethanol manufacture right now. What the byproduct of ethanol manufacture is really pure CO2, and they're learning how to do the CO2 capture thing, working with ethanol plants. But they need fast-growing algae, and those are the ones that don't necessarily make a lot of lipids. And we need better analytical techniques to confirm the real fatty acid content in the extracts that we get. So I've got 3 microalgae that we've worked with, and the total famous mean fatty acid methyl ester by GC. John, in your tour, you saw the GC apparatus where they get the same values. We use Medallion Laboratories, which is a subsidiary of General Mills. They work with foods, and they've done this for years and years and years. They were the ones that really set up this process. And what's nice about it is they have a collection of internal standards that they quantify a test sample against. If there is an unknown peak in there, which could be a fatty acid, they would not have picked it up, but we've had good comparison with them. And we can also determine a weighted average molecular weight from this fatty acid profile we get from Medallion. The gravimetric yield is obtained from an ether extraction, and the FAME determination by NMR, that's what we have just developed. It's a very nice process where you add an internal standard, and it's a simple dimethyl terephthalate. It has a great 4-proton singlet way down field in solution. This is not solid state, this is solution NMR now. And the methyl esters of the FAMEs that are created are in a narrow region right at about 3.28 parts per million. And that's for 3 protons, the other one's for 4. You know the mole percent, and then by knowing the mole percent of the fatty acids in your sample, you can determine the weight percent by multiplying it by its molecular weight. And with denali L-acetylene, we had a lot of, but it doesn't have much for lipids and much for oils in there. And you can see that the extract is much higher than the determined by both techniques fatty acid content. That's because when you extract any living or past living microorganism, you get a lot of stuff out with it. You just don't get the fatty acids. You get the chlorophylls and the sterols and the carotenoids and all kinds of junk. And we did grow a mutant strain of Chlamydia monosterine hardy that had an elevated lipid content at about 9%. And we get, notice again, the extracted yield is high, 12% by NMR. And more recently, and we just got small quantities of this from a company in Austin, Texas called Inspired Fuels that allowed us to publish and mention this data. The highest value we've obtained thus far is 20%, and it's close to the gravimetric. So there's a lot of fatty acids present in this system. But what we really need is a microalgae that has about 50 to really see how good our method is. It's an all-wet process. And this is my last slide. It just depicts the process that we're using. You get 3 products. We take the microalgae, run it through a continuous reactor, right up there, which has an induction heating system. We think we can do this continuously at about 200 degrees centigrade into a filter. The aqueous filtrate can be run right back in with this KNP into microalgal growth. We get a lipid-rich char out of it as a filtered product, which can go through an extraction process to remove the lipids. We call it delipidated char. We knew we made up a word, but it'll work. And the applications in red are the applications that we've really have some data behind. Those in black are hypothetical. They're proposed. And the fatty acids can be plugged right into biodiesel or biogasoline and jet fuel. So that's where we're at. Again, we really love collaboration. If you're interested in collaborating in any way. When I went to that conference at Iowa State, I had great reaction from people. I'm working with about 5 people from contacts from that conference. They were very interested in it. And if you are and what we're doing, I'd be happy to work with you. Thank you very much for your attention.
All right.
Yeah.
With your DDGS, the filtrate,
Yep.
what is the composition there? I know you mentioned some of the composition of the algae stuff.
Yes, and they're similar. It's amazing how similar they are. The chars were identical, and actually we just submitted that for publication. It'll be out soon. I could get you that data completely if you're interested. Yes. 200. Both these were made at 200 degrees centigrade, 2 hours.
When you're recycling the aqueous,
Yep.
your organic solute concentrations will increase?
Yeah, I would. Yes, that's something we have to apply. If you didn't, I'm sorry to repeat the question. The question was, when you're recycling, do components that in your recycle build up in terms of concentration? Absolutely. The Maillard compounds might build up to toxic levels. That's something we need to determine. We have not done that yet. This outfit in Texas we're working with is going to be doing that. Because that's a pretty big thing. If you have a lot of aqueous waste product you've got to get rid of, that's a big deal.
I had one comment on your comparison between use of algae for direct combustion. You had a slide that said you would end up with negative energy. I noticed there that you had taken that contributions of water as a full amount of but most industrial processes try to recycle that piece in many ways. Typically, that energy is a lot less than 100 if you go dry.
Yeah.
So I wondered if you had not doing that.
Yes, the question was, I considered the full evaporative problem with the expulsion of water from the dry microalgae as my comparison. And that we've done that, I admit that. There are ways you can recycle heat and utilize that and minimize that number somewhat. But we also didn't apply it to our system either. But it maybe is a more severe issue with the microalgae.